Everyone. I'm Loïc Jacquemot. I'm a postdoctoral fellow working at the Institute of the Ocean and Fisheries at UBC. I'm working on biodiversity changes in pelagic ecosystem, mostly using molecular tools such as environmental DNA. So welcome to this seminar. I would like to begin by acknowledging that the land on which we gather today is the traditional, ancestral, and unceded territory of the Musqueam people. You may be joining us from traditional lands across British Columbia, Canada, and the world, and we would like to acknowledge that the owners and caretaker of these lands as well. So our today's speaker is Dr. Ryan Kelly. Dr. Kelly is both an ecologist and a lawyer who connects genetic and ecological research with real-world implementation in law and policy, particularly with respect to environmental monitoring and resource management. He's an associate director of the School of Marine and Environmental Affairs within the University of Washington's College of the Environment. And he is also director of the eDNA Collaborative, a group that aims to disseminate, accelerate, and reinforce science that bring environmental DNA analysis and technique out of the lab and into routine practice around the world. So it's very much an area of interest for my work here, and I'm very pleased to have you here, Dr. Kelly. And with that in mind, I'd very much to welcome you, and I leave it to you now.
All right. Thank you very much, Loic, and thank you for everyone for inviting me. So with that, I'll jump in, and I want to start. I was invited to give this presentation, but I want to include Oli Shelton here as a co-author, who's a sort of partner in crime in a lot of, particularly the quantitative work at NOAA Fisheries here in Seattle, and we sort of walk back and forth between offices, and so a lot of these ideas are his, as well as a bunch of collaborators. You'll see their names here. So let me start. This is an overview slide, but is necessary because a lot of people. Some people think about this stuff all day long, every day, and some people do not at all. So just to sort of set our terms of reference here, when we're talking about environmental DNA, we're talking about DNA that's shed out into the environment or existing. So I use the word shed because I think of fish in this context, but it could be any kind of DNA that is existing in the world. And generally, we think about sampling water. Here are some collaborators in Indonesia doing this sampling in the field. Sample a bottle of water, filter the water part out, keep the cells, depending on what kind of filter you're using, but we try and aim at whole cells. And then you've got DNA that you've got cells, you've got DNA you can extract. And at that point, we extract the DNA and you got to make a decision. Are you interested in asking a question like, what's there? Show me many species all at once. Or a question that's more quantitative, like show me how much of this one thing there is. Here I've got qPCR listed, but also droplet digital PCR, ddPCR is increasingly familiar to folks. And so that's kind of, I'm going to break that down. You'll see those distinctions throughout this talk. Like, what are we looking at? A single species? Are we looking at a multi-species situation? They have different statistical properties, different practical applications. And so with that, I think here's the big question for environmental DNA, at least in my mind, that yes, we can. First of all, it is amazing that we can grab a bottle of water out of Puget Sound, and we can see a rockfish, or we can see an orca in our bottle of water by looking at the DNA. That is cool, but it's not actually useful because we knew that there were orcas and there were rockfish in Puget Sound or in the Salish Sea more broadly, right? Like that information itself is not information. It's not surprising. So how do we turn that into data that we can use, right? Insight about the environment. And I think that kind of boils down to this question of, well, what are we really sampling here? And what is it telling us? So a couple of big picture slides here about how we think the world works and how I think we're connecting the world of molecules to the world of, fish or larger bodied animals in the world. And here's one, I think, big picture idea that's worth putting at the very start, which is why I put it, that if we've got species, and those species are made up of cells, and every one of those cells has DNA in it, it must be true at some scale that when you have more of a species, you have more of its DNA. Full stop, right? More of a species has to equal more of its DNA in the world. And then there are a bunch of details that are going to matter here about space and time and about what, if the organism is doing one thing on Tuesday and a different thing on Wednesday, whatever. But ultimately, I come back to this idea that it must be true that when you have more of a thing, you have more of its DNA. And that is sort of a baseline view of the world here. And then at the same time, what we observe is not what's there, right? We have an imperfect observation mechanism, and we've got a lot of noise in our observation mechanism. So what we see, our observations are a product of sort of an integrated product of signal and noise. We can break that down further to think about the signal, the DNA in the world, as being an integrated function of production and loss terms, and then the noise as being lots of noise. In this case, process variability, like is there more DNA from species because there are more of the species over space, for example, and observation variability. For example, our PCR process is maybe not working as we would like, or we have differences among our replicate samples from the world, replicate observations. So this is a diagram that at first seems simplistic, but it actually is pretty profound, because if you treat your observations on the left-hand side as if they are truth, you get nonsense, and that happens a lot. So we try very carefully to think about where our sources of noise are and model those explicitly. And so part of that here's the signal part of that, is we can think about organisms here. Oli gave me tuna, so that's what we're using. We can think about tuna as discrete in the world. You have discrete tuna, and then the DNA is this sort of smoothing function. If you think back to that 2 slides ago of clouds of DNA coming out of every individual, that'll smooth over space and over time. And so you could kind of think of DNA in the water as a smooth blanket of density that smooths over what in organismal terms is a more discrete signal. And actually, the way I think of this is like the Pigpen character in Peanuts, where he's constantly, there's like a cloud of dust around him, and that's how I've come to think about DNA. There's a cloud of dust, DNA around all of us and around all fish and around all kelp, or whatever you want to look at. And then the question is, how does the scale of the DNA signal map onto the scale of the thing that you were interested in? And then often people want to know, well, how does that compare to my net that I drag through the water? How are those things different or the same? And should they be the same? And I want to suggest that probably they shouldn't be the same. So this is another sort of background thought. And then a further background thought is about sampling, right? So you have different sampling frames. Again, could be space or time. And they're going to give you different answers for, do you have a fish in your sampling frame, like in your net, an actual tuna in this case, versus did I detect DNA? So you can look at these different squares as more literal sampling frames. Like over here, you might have fairly little to no DNA and no fish, and maybe you feel okay about that. You're like, aha, my observations of DNA map onto my observations of the fish themselves with a different method, like a net. And something like here, you've got, okay, I've got a fish in my net, or close enough, and I've got a bunch of DNA. Great, that makes sense. Over here, maybe more fish, more DNA. There's more DNA in the sampling frame, but maybe fewer fish in the net. That might be confusing. And then over here, I got DNA in my jar, but I don't have fish in my net. And does that make me question my view of the world? And I think I'm here to suggest that that's actually quite expected. Like you have a smooth process and a discrete process, and you're sampling them in radically different ways. And so they actually should be different. They're providing quite different views of the world. And then there is this largest frame here, where at this largest scale, there should be some very strong relationship between fish and the DNA from the fish. So to give an example of that, this is work that I was not involved in any way. This is Ian Salter's work from the Faroe Islands. Faroe Islands are in the North Atlantic, and these are in green here is the island mass. And it's a great example, but it's a little bit of a sort of a busy figure here. But so the heat map part here is in, I think it's catch per unit effort of cod. And so where the red area is where the fishing industry is catching most of the cod, and then blue, lower catch per unit effort. And then they went out and grabbed discrete bottle samples of water, and those are these circles. And those circles are colored by regions, and I'm not quite sure how they determined regions, but I think these are a priori assignments. And when you look at a site-by-site basis, like a dot-by-dot basis here, there's no relationship between the DNA that they pull out of the bottle and the catch per unit effort in the net. And this is very common in DNA surveys, eDNA surveys, right? Where you say, okay, I dragged a net through the water, or I visually counted things, and then I did it with DNA, and they didn't match. Like something must be wrong, or it's not working. And very often that comes from this mismatch of scales in space or time. And in this case, it seems like the DNA is smoothing over different scale than the nets are in the water, because when you look at the regional scale, they're very well correlated. So how I interpret this, at least, is that these things tell us, this difference in correlation is telling us something about the spatial scale over which DNA is smoothing the signal from the cod. That in turn might inform like how much you want to sample and where, because you've got a fundamentally different tool for seeing the world, and you really can't expect it to work in exactly the same way as the existing tools. So that's one example. Now back to a little bit of intro, and then I'm getting to a lot of really great stuff coming out from postdocs mostly and staff scientists that I work closely with. Here is, misspelled Ole Shelton's name. That should be Shelton. Here's an example of the kinds of data you would get from a qPCR assay. Single species, this is Chinook salmon in Skagit Bay here. These are endangered under the US Endangered Species Act, and sane biomass. So here it's a little bit of a noisy relationship, but it's they're strongly correlated in terms of the DNA and the sane biomass at the scale of the whole bay. They're not strongly correlated at the scale of individual samples, just like Ian Salter found in the Faroe Islands with cod. So that's the kinds of data that you would see with qPCR or ddPCR, single species assays. The kinds of data you see from a multi-species assay look generally like this. People use these stacked bar charts to show multi-species data where you're looking at, in this case, these are all families, and stacked bar, it doesn't actually matter what these particular ones are. These are samples of different space in the Hood Canal here. But the point is to get a sense of the visual of that's the kind of data that comes out of a multi-species metabarcoding assay. And we got two important notes on metabarcoding before going any further, because this causes great confusion and anger, I think is maybe not too strong a word. So first of all, the first thing to know about metabarcoding is you're amplifying a mixed template, right? You've got a sample from the world. It might have 1,000 species in it. It might have 100 species of vertebrates, let's say, and you care about vertebrates and you're going to amplify those with PCR primers, but they're not all going to amplify identically. The reason for this is differences in primer template binding and maybe some other things, secondary structure and so forth at the molecular level. But what that means is you imagine PCR on a log scale here, log copies and PCR cycles on the x-axis. And so at a log scale, this is going to look linear, and you've got some number of copies of amplicons that are building up as you go through the PCR process. at time zero, at 0 PCR cycles, is the number of copies that were there before PCR. So in a mixed template, you've got these different species. Here in this visualization, dark blue is this minority of them, but it amplifies slightly better, slightly more efficiently than the other species, such that after 30 cycles of PCR, it actually is now the majority of sequences that you're seeing out the back end of your sequencer. So the mistake that people routinely make is to expect that the things coming out your sequencer in these proportions should look exactly like the proportions that went in. And it's not true because they amplify at different rates. So you have to model that explicitly. You have to ask, well, how well does each of these species I care about amplify? And then given that, you can correct for it very well. So that's this 2022 paper that gives the math for how to do that. But if you look, these gray dots are the scatter plot of expected versus observed without correction, and then expected versus observed having been corrected, they fall right on the line. So this is very explainable, very achievable. You just have to know to do it, and it gets rid of this problem of differential amplification. The other important note about metabarcoding is what to do with the data once you've got them even corrected, right? These are compositions. And very often people will treat them not as compositions, they'll treat them as sort of independent, the species independent of one another, and that's not true. So you've got, if you want to treat them independently, you've got to have some additional source of information to get out of composition space, out of proportion space, into absolute concentrations. So you could do that, or absolute, abundances. And I'll give a couple of examples of how one might do that, but you need to pair it with things like, visual counts or trawls. You can also pair it with other molecular data, like a qPCR assay or ddPCR assay. That has absolute abundance in it, and then that can break out of composition space, and then you can be sort of dealing with more standard statistics. But what we do is model the PCR process for each species in the composition, and then you can do that with a softmax transform, and then we imagine that our observations are subject to some noise from this multinomial sampling that is happening at the sequencer level. So, okay, all of that was a lot of like, you got to eat your vegetables before we can sort of dive into the good stuff from field work. But that's at least sort of sets out how we've been thinking about the world of eDNA and how to make sense of it. So let's now look at a bunch of real-world applications from species that perhaps you know and love, are nearby. A couple of single species examples, and then 3 multi-species examples. And I want to just sort of highlight that these are all different combinations of data sources and slightly different, questions or parameters of interest. So that will be clear. But each project involves, obviously slightly different aims, and so as a result, we can use slightly different tools to get at those parameters of interest. So first one up here is about the single species and thinking about abundance, how it maps onto abundance of a species that we care about in the world, and whether or not the DNA is going to accurately reflect that. And hopefully I've convinced you that I think there are good theoretical reasons why it should, and there are good theoretical reasons why that goes wrong a lot with sampling. But here are 2 radically different scales. And this is Hiroki Yamanaka, who is visiting my lab actually for a year on sabbatical from Japan, and Toshiaki Joe, who have a lot of great work out of Japan. Here is a highly replicated study of zebrafish in small aquarium tanks over and over again. It doesn't matter what filter pore size you use. Those are the different results in columns here, the facets in columns. It doesn't matter what DNA fragment you use or the size of that fragment. Those are the different rows here. You get a very, very good relationship between the number of the biomass of fish in the this is abundance. The abundance of fish in the aquarium and the DNA that you're seeing out the back end. To me, the lesson here is you at a very, very small scale, like you're sampling that universe very well. And so of course there's a good relationship there. However, at much larger scales that often we care about in ecology or here in fisheries, that wasn't true that was going to hold. And so this is a study that took a long time to do. This is the 2019 Hake survey along the west coast here of the US. The Hake survey is an acoustic trawl survey that goes along these transect lines all up and down the coast, doing acoustic trawls. And in 2019, they very kindly took bottles of water for us, which are these red dots. And then, after a lot of analysis later, we get this plot. This is the eDNA index as derived from qPCR assays for the Hake. This is actually flattened into 2 dimensions so that it could be easily compared to the acoustic trawl survey, which is here on the right. So first thing you might notice is they look like each other. So that's great. But the other cool things that came out of this are, well, number one, that it seems like eDNA is giving us a very good sense of where the fish are. But as I said, this has been flattened into 2 dimensions. We have it in 3 dimensions. These were sampled down to 500 meters in depth slices. So you can actually get 3-dimensional views of where the fish DNA is at large scales. And I'll show you another example of that coming up. But I love that. The other cool thing here is it's very easy to replicate, right? You can take 3 bottles of water instead of 1. It's not a lot more effort. You can do triplicate PCRs in the lab. It doesn't triple your cost. And so you can very quickly get a sense of your signal to noise ratio and your coefficient of variation on your observations in a way that becomes very hard to do with acoustic trawls or other methods that get used at large scales. So take home here, this is single species work at small and large scales. As long as you're sampling at the appropriate scale in space and time, you expect a good relationship with biomass or counts of species, and indeed you see that. Here's a cool example. This is Abby Keller's work. She was a master's student with me at the time and now a PhD student in Berkeley, and has this R package called eDNA joint. And this is for developing Bayesian joint models for single species work, which might be useful to some of the folks in the audience, where she had information from traps, traditional traps, where it's trapping an invasive green crab, that same one you guys have across the border, Carcinus. The Washington State Department of Fish and Wildlife put in a lot of effort trapping these things, counting them, so they knew where the crabs were, how many they were. And Abby went and took DNA samples from water alongside those traps, and then made this. And so the yellow dots is where she found DNA and no adult crabs in the traps. The purple dots were where the traps and the DNA agreed, in that there was an adult crab in the trap and also a positive hit on the DNA. And then the gray was neither. And most people would stop at that point, and say, that's what I found. And they disagree in some places and they agree in other places. But Abby said, no, I've got 2, imperfect views of the world. I've got one from a trap and one from qPCR. Can I make a joint model to estimate crab density in equivalent units of crabs per trap, and then apply that over the space that she sampled? And then I just think this is so cool, because it ended up being an R package. It ends up being useful for lots of reasons, but mostly because it can speak the language. It puts molecules in the language of crabs per trap, which is what the management agency in this case, that's how they count crabs. And so it sort of makes it a more immediately policy relevant measure. So I thought that was great. And then you've got a couple of instances here where the DNA showed up and the crabs didn't. And we did enough negative controls that we could, show pretty well that the likelihood those are false positives was really, really low. So what we think that means is that, well, we're seeing larvae in the water of the invasive species long before you're seeing the adults, which makes sense as a matter of biology. And it also points to the management agency, say, hey, here are some places where you might want to put more effort into looking and making sure you could sort of get any of those individuals if they show up early detection, which is, economically, obviously much more efficient than later detection. So then now switching from, so those are a couple examples locally of single species eDNA work in the context of management, and in particular quantitative work. And here we're going to switch over to multi-species metabarcoding work, where we're looking at lots of species at once. And there you've got this problem of proportions. And what do proportions mean? How do we handle them? How do we make sense of them? Here, this is Gled Gori's work. And Gled was a PhD student in Tromsø in Norway at the time. He's now here with me as a postdoc, and you'll see more of his work in just a second. But Gled has this paper that came out last year in IC's Journal of Marine Science, where he's using trawls. This is trawls in Norwegian fjords, and it's about 5 or 6 species of fish that are commercially important, caught in trawls. And so Gled had the trawl catches from Norway over some number of years, 3 years to be specific. He also had a single species assay, a ddPCR, for more than one of those species. And he also did metabarcoding using primer sets that amplified the fish in those samples. So he had proportions from the metabarcoding. He could correct them appropriately for amplification bias because he had measured that. He has absolute concentrations from ddPCR for some of those same species of fish. So he's got molecules in 2 different ways. So in the DAG here, in the model diagram, you've got molecules in 2 different ways. Those molecules are both speaking to some parameter estimate for the concentration of species-specific copies of DNA present in the environment, or equivalently here, present in the DNA extract coming out of the water. And then he's got observations from the trawl model. So he knows how many showed up in the nets. He knows the variability around that within a year and between years. And then he jointly models some true fish density in the world, given the trawl, given the DNA. And this also allows him, then what falls out of the model as kind of a bonus is the relationship between these things. So species-specific, I'll call these hypotheses about how much DNA is there per fish in reality, per fish in the trawl, and so forth. So we put all this together, really impressive piece of work over about the course of about a year. But what's most impressive here is that he could train the model on 2 years worth of data and estimate the 3rd year of data as an out-of-sample estimate, and then do that for each pair of years and estimate the 3rd year. So sort of a round robin out-of-sample cross validation. And he could predict the number of fish, the fish catch in the trawl from the molecules alone. Having trained the model on 2 years, in that 3rd year, he could predict how many fish would show up in the trawl and do that quite well. So there were a couple lessons there. First of all, when you have very rare things, don't work at all. And that is really obvious in that paper. When you get very few observations, there's no real signal there, so it won't work. But for fish that are pretty common, which are the fish that you're commonly fishing, it ends up working really well. And now the Norwegian government has followed this up, actually, and now there are 6 years worth of data, and they really want him to refit the model and see if it falls along the line, because increasingly it looks like if those parameters remain constant over time, it seems like you literally could count fish in this case from the molecules alone. You can't do that without a lot of math. You can't do that without the trawls in the first place to help you understand what your relationship is between the molecules and the fish in the net. And so what I'm not saying, I'm emphatically not saying that you could just go grab DNA from the water and count fish immediately with no other information. But what I am saying is that when you have a rational framework for understanding how the molecules are relating to the estimates of fish in the world and the estimates that you're observing from your trawls, then all of a sudden it does start to make sense, and you can start to make equivalencies there. You can say, hey, what information am I getting from each part of my model, and can I optimize that, right? Can I do some number of fish trawls and some number of bottles of water for DNA and put those together in ways that give me the optimal amount of information per dollar spent, for example. And I think there's no reason not to do that. But I definitely want to underscore that I'm not saying you can go grab water and count fish just from the molecules with no other information, because I don't think that's true. So that's Gled Guri's work from the Norwegian fjords. You'll see more from Gled in just a second. This is a quite different example, but quite similar math. It's an example where you've got metabarcoding proportions, again, corrected for amplification bias, and a visual survey from Hawaii. This is the Big Island of Hawaii, the west coast of the Big Island of Hawaii. The NOAA office in Honolulu spent a lot of time and effort swimming transects over the reef, visually counting fish, and while they were at it, grabbing bottles of water and grabbing DNA. And they call that visual survey, visual census, UVC, for underwater visual census. And then Oli built this joint model of the DNA and the visual survey, just very similar to what I just showed for Gled Guri's model in Norway. And here's what we get out. We call this lovingly the Atari plot, because that's what it looks like to us. And when the darker colors here, black being the extreme example, are unobserved in the visual data, and then the warmer colors are more commonly observed in units of kilograms per, I think it's kilograms per transect that the divers were swimming. So this is what the visual survey looks like. Here's what the joint model with DNA and visual looks like. And so what you immediately see is that this thing lights up, and you're instantly with DNA, you're seeing a lot more stuff than was there before. And so that's one thing you might immediately take from this, is to say, well, when you look at two sources of data, you see more than one source of data. And that's trivially true. That is perhaps trivially true. But what's remarkable here is these are bottles of water taken alongside the divers, and like, what are the species that pop out more? It's a lot of them, if it was common in the visual, it's going to be common in the DNA. That's great as a sanity check. But then there are things where you didn't see them at all. Like this species here, is this Encrasocalina? Yeah. Here you have zero observations in the visual. And it is, I think it's actually the most common species in the eDNA. And what's going on there? Well, that species, it's an anchovy, and they form large aggregations. They swim in and out. They're kind of on the reef, but kind of not exactly on the reef. But you would never see them swimming a transect, looking down, writing down what you're seeing. And that's why there were zero of them in the transect. This actually is one of the more common bait fish there is in the Hawaiian Islands, a very high value bait fish. And it's totally absent from the traditional survey. But the DNA picked it up and accurately decided that it was, that it was very common. It was the most common fish there. So this is a great example to me of like, what holes what are you not seeing when you're using any one method in the world? You're not seeing a lot. And so the DNA allows us a really different view of the world. And then what we try and do is wrestle with putting together competing views of the world, so they're not competing anymore, that they form a more unified whole in a joint model. There are a couple of other examples like that. So some of these are of conservation, real conservation concern, and here's one of them, the ulua, giant trevally. This is the kind of thing that it was unobserved in the visual census. However, you know that there are some of them there. It's just not the kind of thing that shows up on a visual census. It's a large body thing, and there's, it might zoom through, and it doesn't get seen on the visual survey. But the DNA is certainly there, and then in some places it's quite common. So that and then you can map that onto space and say, here's where we think the ulua are hanging out. Here's where there are more ulua than fewer. That turns out to be exactly what the NOAA office in Honolulu wants to know for for management, cultural, and conservation reasons. So all kinds of good stuff coming out of this. While I'm on this slide, I also highlight that a lot of these species that they're not seen at all during the day, but we see a lot of them in the DNA. It's because they're nocturnal. So you do a visual survey during the day, you don't do it at night, and then you get to see all the DNA is still there even during the day. So that's cool. Also, small cryptobenthic reef fish, right? Things that would never show up when you're looking with your eyeballs, but the DNA can't hide. So we're writing that up now. John Whitney is leading that work out of Honolulu. And then finally, last example, this comes from the same set of samples, those 2019 Hake survey samples from NOAA, where we've been reusing those in various ways. We've got a multi-year marine mammal project that uses those same samples. We can go back and requery them with different primers, look for different species. Here, same guy, Gled Guri, has taken this and run with it with the metabarcoding. So instead of single species, just Hake DNA, here we're looking at the DNA for lots of vertebrates. And here's an example of 6 of them. And let me orient you to this. So the species are labeled. The facets here are the depth bins that I alluded to earlier. These are depth bins at which NOAA grabbed water with a CTD. And so we're getting a 3-dimensional view of where, in this case, anchovy DNA is across, along the west coast of the United States over 10 degrees of latitude. And then Glad, in writing this paper, which is currently in review, he's broken the fish into sort of different habitat types: surface, midwater, and deeper water fish. But take a look just at anchovy and sardine. You can see the anchovy are located a bit further north, the sardine a bit further south, consistent with what we know of their ecology. And every time I see this, it blows me away that here we have over 10 degrees of latitude, we have a heat map of where the DNA is, and therefore where the fish are, without ever seeing a fish, without ever counting one of these. And that just is completely remarkable to me. So one question's about the depth resolution that we can get, and here is a great example. This is the widow rockfish, and here we see its DNA at 50 meters and really not elsewhere. And so that's an example of where that gives us a lot of confidence that it's not that we're just seeing DNA sort of all things everywhere, but it's here quite restricted by depth bin. There are some species that are deeper water fish, like this is a dragonfish, I think it's called, where yes, the high point is at 500 meters, which is what I would have expected. But then there's a weird aggregation at the surface here in the north, and like what's going on there? And it turns out there's a larval stage that lives at the surface, and so we think probably that's what's going on there. Other things like Stenobrachius, this is a fish that engages in diurnal vertical migration, and so you might expect to see the DNA everywhere over the course of a day, because the fish is kind of everywhere over the course of the day, and that is what seems to be happening there. So I just love this example of being able to take, first of all, samples that were taken for one purpose, looking at Hake, and repurposing them for completely different things, looking at, I mentioned marine mammals, and this is, these are different species of fish that some of which are commercially important, others of which we just don't know a lot about, but it seems like there are a lot of them, particularly the deeper water, fish, the myctophids and so forth. And then mapping them in a way that seems very much like this is mapping the fish without ever seeing the fish. So to me, that's really, really exciting, and it's fun to have an opportunity to get to work, really what I feel like is at the boundaries of what's possible, and push this into, into practical utility at scale. So that's super fun. Some of you will note the scale bar here. It is quantitative in the sense that it's quantitative as to DNA. It's not necessarily quantitative as to fish, right? Copies per microliter. So if people are interested in doing stock assessment, I can't tell them copies per microliter, because it's not a unit that makes sense to lots of people. And so then I think there is the next step is to link it to other methods of seeing the world in just the way Glad did in that dissertation work, so that you can develop those kind of equivalencies and translate between units of molecules, units of fish, and make sure it's speaking to whatever the need is, be it a sort of research need or management need. So I'll stop there because that's a lot, and I wanted to leave ample time for questions. I realize also I've been speaking very fast given the amount of coffee I've had this morning, so I'd like to apologize retroactively. But eDNA work is a real team sport because you've got a lot of field work often, you've got a lot of lab work, you've got a lot of math, you've got a lot of graphics and writing and so forth. And so I'm sure everybody didn't make it on this slide, but truly this is an enormous group effort to have come up with what I hope were interesting examples that these folks have worked hard on over the last few years. And yeah, I'm happy to let's take questions now.
Yeah, thank you, Dr. Kelly. That was a really interesting talk. We have some people online that I'm going to be monitoring the chat for questions. We also have a whole group of people at the university in one of our seminar rooms. So we'll probably get some questions from the floor. We'll see how well our microphone works. I may have to repeat it. While we wait for some questions on the floor, I'm going to use my prerogative to start with a question. And that's going back to the very start of your talk when you talked about the amplification process. And as I'm peripherally involved in changing genetics into quantities, the one thing that as a non-geneticist person, I always imagine this black box, which is the amplification process. And I always assumed the genetics people had a standard black box that they bought off of BWR or something. And when I started getting into it, I realized there's a lot of variability in the data that you get out of the amplification process based on the methodology and the equipment used. That was several years ago. Is that still a concern, that transferring those genetic samples into genetic information is highly dependent on the methodology in that process?
It's a great question. I think it depends on what you mean by the methodology. So it was definitely, there was a time, for example, in the 90s with microsatellites where like you would run the same sample on two different microsatellite machine, two different sequencers and get different answers. And that's untenable, right? That's no good. I think that is no longer the case, right? If we do the same lab work, we're amplifying with the same primers, and we put it on sequencers, we'll get very similar answers. So in that sense, as a technological matter, I think all of that has stabilized quite well. However, if you amplify with different primer sets, you'll get entirely different answers. Because you're asking entirely different questions of your data. So that's a thing where it's like a subtlety where it depends on what you mean by the methodology. Like if we were following the same protocol, using the same tools to ask the same questions, we're gonna get the same answer within a very narrow tolerance of technical variability, and we measure that. If we're asking different questions with methods that both happen to be molecular, then we're going to get radically different answers, and we just have to be clear what we mean when we're saying, hey, we're using molecular data here. Does that answer your question?
Thanks. Yeah, it does. So it's still, it seems to be that the exact primer methodology is key to understand and to express properly. So
100%, yeah. And in fact, that's when, it's nice working with NOAA because they force us to sort of speak in more practical terms a lot of the time. That at one point they sort of looked at us and they're like, oh, the primer set's just a different net. And I like that's sort of landed really well, that different primer sets are different nets. And some of them have big mesh size and some of them have tiny mesh size, and they're aiming at different things. Just like you might not say, hey, I dragged a net through the water and this is what I found. You might say the net matters.
Right. Let's start with some questions from the room. Go ahead.
Hi, I was really curious when you were showing all the very beautiful maps of how currents influence fundamentally your eDNA space, and then how that limits how you interpret the data kind of between different geographies.
Yeah, great
So
question.
how, like a model that's in one fjord, would that translate to the fjord next door or not, for example?
Can I just ask, could you hear the question from the room okay?
Yeah, I think I could hear it great. I'm not sure I would know otherwise. We'll see. Yeah, no, we think a lot about that, about sort of transport of DNA over space. And so let me actually go back to that slide just as one example. Is that going to work? This. So here, this is a smooth, it's a spatial smooth, and kind of the question is like, is that an accurate representation, or is that on one day? And so like my favorite version of this question actually is, how do you know you're not looking at dinosaur DNA? Because it's actually kind of two questions, like how old is the DNA, and then how far might it have traveled? And so we've tried to wrestle those questions to the ground. The half-life of DNA in the environment like this, like in ambient, temperatures and seawater, you're looking at a half-life of like hours to days at most. And so that limits how far it can travel. And the distance in this particular survey, the distance between survey points was much greater than the distance that it was likely to travel before it degraded. And then there's the signal and noise ratio question also, that as you travel, you sample one place to another, the vast majority of DNA you're seeing in one place came from that place. And yes, some of it is transported from elsewhere, but at very, very diminished rates. So it's a tiny fraction of things that you would see in the first place, even if there were not much degradation. And then there's a lot of sinking that happens, or other ways that it sort of doesn't get detected. So what becomes really clear is that these are that our sampling scheme in this case is larger. Our sampling points are further away than any given molecule is likely to travel between them as a result of transport. Does that answer your question?
That's really helpful. The second part of my question was how, for example, you showed the where you kind of turn the eDNA into graphs per track. To what extent could you use the same model that you developed in an adjacent space that wasn't part of the model space?
Oh yeah,
How much
sorry.
you got that?
And yeah, thanks for the reminder there. And this that slide, this one, this one has no spatial information in it. These are just one-to-one on a site basis. So this doesn't have any oceanography, it doesn't have any transport in it. What I think we've learned about transport is that the vast majority of the transport of DNA is going to be driven by oceanography. So if you've got, it's the velocity vectors that will do it. And in some places, tide is the thing that's going to transport stuff in and out over the course of a day. In the open ocean, it's probably not that. But if you have the velocity vectors of, the kind of gross oceanography of the place that you care about, that gives you probably a very good first order approximation of where the DNA is going. And then if you wanted to sort of ballpark it, you could say, hey, if like where was this water mass within the last hour or two? That would be my first guess. So it's not that just like every oceanographic model isn't going to apply to a different place. But the terms, the like decay terms, the understanding of sampling, the understanding of what we're seeing when we amplify and sequence DNA out of the world, all of that is very general. And then as transport is always, I think, a very particularly local question.
Super, thank you.
Okay, I'm gonna take one of the online questions, and one is getting back to the methodology about the PCR amplification process. And if I could summarize the question, if you get the amplification efficiency out of a sample or a mock community, does it maintain that same efficiency coefficient in another sample of different composition? So
It does.
The composition effect, okay.
Yeah, exactly. So that it's a function, thankfully, or else this wouldn't work. It's a function of of the primer binding energy between the primer and the species. So it's independent of the composition of the community, and it's independent of you know, the proportion, the composition in two different senses of the word, both the other species that are there and in the proportion of those species. And we've, you know, cross-validated that in many different ways to convince ourselves that that was true. So that amplification efficiency is portable in that sense.
Okay, switch to, is there a question in the room? Go ahead.
So I would like to know if there are like conditions that are better for this type of technique, like for example, the air, the area, the temperature of the water, and if also you have different answers in a temporal scale, like from summer to winter. So how the technique goes with these difference?
I don't know if you could hear that, it sort of came from the back of the room. I guess the
Yeah.
question is, what kind of oceanographic conditions affect the efficiency of the answers, and that includes seasonal effects?
Yeah, the thing that so I mentioned currents as, you know, that's sure, like DNA definitely gets transported by currents, but it doesn't go as far as you would think. It doesn't last as long as you might think. In a river, for example, DNA does flow downstream, but it doesn't go many kilometers. So it's interesting. The other large scale thing, the kind of big picture rule is that the DNA is actually getting lost because of biology. It's being eaten by microbes, and it's being, you know, it's sort of degrading in the environment in a way that is dependent on the biology of the system, not just on sort of chemistry or physics. And so at higher temperatures, DNA degrades much faster. So that is a good rule that, like, you would expect if you're in the tropics. I would expect that DNA would not effectively travel very far at all because it's degrading much faster. And in the Arctic, it should last a long time. You're basically, you know, living in a refrigerator. So that's how we think about it. That's how it plays out. We can also use markers. It's not in this talk, but we can use the fragment sizes of DNA as a marker of time. How long you know, a given cell might have been in the world is actually is predicted by the fragment sizes of the DNA, because it's being fragmented. So the small fragments last longer than big fragments, and you can use that out of the same sample. You can look at a variety of sample of fragment sizes, and then actually triangulate backwards to ask, well, what's the average age of the DNA in this sample? And that's Pedro Rendau's work that came out earlier this year.
I know we have a number of graduate students working various aspects of eDNA, and there's a very simple question, maybe has a simple answer, but maybe not, that you can answer to help them. And they had a question based on one of the maps where you had converted the 3D data into 2D. And they were just wondering whether during that compression, is it straightforward as averaging all the data, or is it something more nuanced, and maybe some you can suggest, if it is more nuanced and complicated, perhaps you can provide somewhere they could look to understand that process more.
It is an easy answer in the sense that I don't know the answer. So I'll have to direct you to Ole Shelton. I believe it's just the sum over all the depth layers, but we'll have to check with Ole on that one.
Good. And similar to that, can you recommend any good resources or comment on eRNA stability in the water column? How much is known about this?
Yeah, we've been doing a bunch of work on that, and actually, Pedro Rendau is the first author, and it's actually the same paper. I can't drop anything in the chat right now, but I can tell you, Pedro Rendau Diaz, and he has a paper in Scientific Reports that came out about 2 months ago on age in DNA samples, fragment sizes, and RNA. And so with RNA, you've actually quite different molecules that, like, messenger RNA is a really different thing than ribosomal RNA. The ribosomal RNA is going to behave like DNA, it's going to last a while in the environment, you know, days, let's say. Messenger RNA goes away very, very quickly. And so the timescales are going to be much shorter with messenger RNA. So if you're looking for like functional genomics, you're really looking at a very narrow snapshot in time. And that's Aidan If, who's a staff scientist here, is working on that work actually in salmon, where you can grab water out of the river and look at what genes are expressed by looking at the mRNA that you sequence out of that river water, and whether that maps, you know, in time on what we know is going on with the salmon run. And sure enough, it is. You can see sort of spawning genes peak at the appropriate time. You can see sort of genes associated with decay and degradation of the, you know, the individuals as they're dying and hanging out in the river. So it is possible to do mRNA work like that, but you have to just be prepared for the idea that it's like a snapshot that's not more than a couple of hours at the very most, because it decays so fast.
Thank you for a really engaging talk. It's interesting seeing it from all aspects, from the technology to the mathing to the actual, some of the results you get. I'm sure you'll get many follow-up questions from the audience, but in the meantime, my great thanks for a really interesting seminar.